Okay, so we're okay. We're okay to start now. I think we have enough persons. So again, welcome everyone to our Bible Paper 2 Marathon. My name is Jhada Haughton and I will be your tutor. I can't really turn on my camera right now for the sake of not having everything fall off because I'm having some Wi-Fi issues. That is why we started late. But outside of that, we should be fine. So I'm gonna start with just a brief introduction. I'm not sure if everyone had heard, but in regards to the grading, our labs are worth 20%, paper one is worth 30%, and paper two is worth 50%. So we are working with paper two, which has the bulk of the information, or the not the information, the bulk of the grade. So paper two is composed of 6 questions, and all 6 of those questions are compulsory, unfortunately. You will not be able to choose which questions you want to do. So you have to have a good understanding of most topics going into the exam. So that is what we're going to be trying to expose you to as much content as possible. I had sent from paper 2017 to 2023 papers in the group, and I had asked persons to start looking through and just jotting down some answers so we don't spend a lot of time in silence waiting for everybody to come together and come up with an answer for the question that we're going to talk about. So I hope everyone had done that, or you had started to at least look through the paper. So we're going to be starting with 2023 paper. And please, people, I am a teacher. I don't like when I am teaching or when I'm doing something that is like I'm talking to crickets. Everyone is here to participate and everyone is here to try and get something out of the session. It makes no sense you sit there. If you're not comfortable opening your mic and saying something, you don't have to do that. You can message in the chat. If you want to speak, you can raise your hand and you will be acknowledged to give your answer. If you have a question, that will work as well. So please don't just sit there and don't say anything. This session is for you, and your participation is needed to make it go smoothly. Right, guys? And you can just send a thumbs up or something, or say yes in the chat or something, so that we're all in agreement that that's what we're gonna do. All right, Crystal, CEO, yes, Justin. Who was that talking? Chantel?
Yeah,
Or
someone is
Chanel,
saying that
sorry.
someone is saying that they can't hear you properly.
Can't hear me. All right, let me try something. Is that person, is everybody else hearing me correctly or clearly?
Yes, miss.
All right, so I'm not sure if that person, if you're not hearing me, maybe you need to rejoin the session or move to a new area because other persons are hearing me clearly. All right, so I'm going to start sharing my screen. As I said, we're going to start with paper 2 from 2023. Is everyone seeing?
Yes, miss.
Yes, miss.
All right, so again, on the paper, the first thing you're going to see are instructions. 6 questions in 2 sections, section A and section B. Section B usually starts from about questions either 4 or question 5. You either have 3 or 2 questions in that section. Those questions are usually essay type questions, so you have to write more information versus section A, which it is still questions that you have to write sizable answers to, but it's more in the short answer question range, right? So you have 6 questions, all of them are compulsory. I believe that you should be allowed with a calculator in the exam because they don't specify that you cannot carry a calculator. The reason being is because sometimes you may have to work out something. If you are given a drawing to do, you might have magnification to work out. And if you're going to plot a graph, sometimes you would like to work out the scale. So if it is possible, please walk with a calculator to the exam. Please also ensure that you walk with a ruler. Again, if you're given something to draw, you will have to abide by all of the guidelines that you would have had for regular biolabs, where you would have to draw your label lines with a ruler, and you would have to have your title underline, all of those things. So work with a ruler and a calculator into that exam, please. Pencil, if you can get a mechanical pencil, that would be good because that is better for cutting graphs, and it's also better for when you are doing your drawings. If not, then just ensure that you work with your regular pencil and you have your sharpener, so you're able to pay attention to that. So we're going to start. The first question on the paper is usually a lab-based question. So it is going to ask you something based on the labs that you would have done, or it is going to assess some type of laboratory skill, where it is going to ask you to either draw, it's going to ask you to give an example or to describe a lab scenario, or to give you an actual lab and ask you information based on that. So it is always a lab-based question for question 1. You also will sometimes be given graphs to either draw or to interpret, but anything that falls under the lab skills, that is what will be in question 1. Understand that, guys?
Yes, please.
Yes, miss. Yes, miss.
All right. So that is question 1. So the first question for 2023, you see that you are given micrographs of an animal and plant cell. And it's sometimes it's not really that clear, but you are able to see enough details. And it asks you what type of microscope was used to produce the images shown in figure 1. What kind of microscope was used?
An electron microscope.
Okay, so we use an electron microscope. How you can kind of figure out what you're using. The electron microscope, they usually call them micrographs. So that gives you a clue as to what kind of microscope you're using. They're only ever going to ask you about electron microscopes or light microscopes. And we know that the difference between the two of them is that the light microscope, it magnifies things by times 10 to about times 1000. And obviously, because it is a light microscope, it uses light as its means to basically view the specimens, versus an electron microscope. What is different between the electron microscope and the light microscope? Can they see the same amount of detail, or can one see more? Anybody? Somebody? All right. I guess no one knows that one. But your electron microscope can see smaller details. The magnification is in the thousands. And as I said, it's always called a micrograph. So you can also just look at it in general. If it is a light microscope that the picture is from, it will look different from electron microscope. the electron microscope usually uses contrast, so you have very dark areas as opposed to very light areas. So this is obviously from an electron microscope. So that's how you would identify it. Are we clear, guys? Good with that one?
Yes,
Yes,
miss.
miss.
All right. And then when you go down now, it says draw a large clearly labeled diagram of one plant cell from the micrograph shown. So again, I said that this is a lab-based question. So when you're drawing this specimen, you have to abide by the rules that you would have used for a regular laboratory drawing. So who can let me know some of those things that you have to abide by when you're doing a drawing for a
All
lab? What are some of the things you have to do?
all labelings are labeled to the right or something.
All labelings to the right, all labelings and annotations to the right. Yes, what else?
Title at the
No
bottom.
double lines. Yes, no double lines. So you're not supposed to sketch it. Your title is supposed to be at the bottom and it's supposed to be underlined. Some
And
persons will mark it for it being in capital letters, while some don't mark it for that. But for your safety, do the title in capital letters. And your title just describes what you are drawing. Someone else was trying to say something. You can go ahead.
use a sharpened pencil.
So you'll use a very sharp pencil. Hence why I told you guys that when you're going into the exam, you either walk with a mechanical pencil because the lead is always one size, so it's easier for you to draw with that. And if you have a regular HB pencil, you have to ensure that the point is very sharp and thin, so you don't end up with lines of different thickness. So all of those things that you would have used when you were drawing a regular specimen in the lab, you have to use that for this. Again, that's why I told you guys to work with your rulers as well. So if we look at the micrograph of the plant cell, it's not very clear, but you can kind of use common sense and figure out where everything is. So we would have our cell wall, obviously out here. And then this faint line inside of that would be your what? What would that one be?
The cell membrane.
Cell membrane. And then in here would be obviously a cytoplasm. Or well, from this one though, it would be safer to say that this is the vacuole because the vacuole is usually located in this area. Then you would need to have at least your chloroplast and then your mitochondria and your nucleus. So where would be the nucleus in this one? Just give me a vague idea of where it would be. So you can tell me if it's to the right, if it's to the left.
To the left.
To the left. So it's this thing up here. You always can identify the nucleus because sometimes it will look granulated like it has little dots inside of it. And obviously that very distinct nucleolus, which will take a lot of, if it is a regular image from a light microscope, it will absorb the most dye, which from whichever dye that you're using, it always absorbs the most, so it will look the darkest. And in this case, we can still see it because it shows up as that little dot. So this is our nucleus. And then these dark structures that you see, these are your chloroplasts. Anytime you're looking at electrographs or even sometime in the light microscopes, you can kind of see the inside of the chloroplast where it looks like it has some striations. It kind of looks striped because it would be showing you the grana that is inside of it, and they're always stacked on each other. So if you're trying to identify the chloroplast, you can look at that to try and identify it, and it is usually one of the larger structures. So that's our chloroplast. And then these faint dots that you see are your mitochondria, right? So those are what the different things are. So again, just ensure that you can give a cell wall, the cell membrane, your nucleus, and your chloroplast. If you cannot identify anything else, you need to be able to identify those. The cell wall is always the first outer line, and then whatever line is contained within that, whichever boundary comes after that, that is your cell membrane. The nucleus is always going to have that dark spot there, and then your chloroplasts, as I said, they always look like they have some little striations, and they're usually one of the larger structures that are inside of it. And they're usually like shaped like cylinders, elongated. So those are how you would identify them. And when you do your drawing, you ensure that you have your title underlined at the bottom, and then you have your magnification. How do we work out magnification?
Drawing size over actual size divided by actual
the
size.
Drawing size divided by actual size. And when you work it out, it won't have any units because the units will actually cancel each other out. So whatever you got, if you got 10, you're just going to put times 10. Because it's telling you that your drawing is 10 times as large as the actual specimen. So that is how you would do your magnification. Always ensure that you show the working out for your magnification, and that you actually show the you actually put the magnification here beside the title. So those are the guidelines that you would use when you're doing the drawing lab. And again, all of the structures that are there, at least ensure you have your vacuole, your nucleus, your cell membrane, your cell wall, and your chloroplast, if it is a plant cell. For an animal cell, you would really just need to ensure that you have your cell membrane, your nucleus, and maybe if you want to indicate the cytoplasm, you can do that as well. So set cytoplasm, cell membrane, and nucleus for the animal cell if they had asked you to draw that. We're still okay?
Yes, miss.
Yes, miss.
All right. And you see how much this is worth? 9 marks. So it's question one kind of easy when you look at it. Whatever they're asking you is a lab skill, it's not usually too hard. So even if you can't do the entire question, there are various parts of the questions that you can actually do to get a lot of marks. Because you already got 10 marks out of the question already. So you answer what the microscope that was used, and you would have done your drawing. And once you abide by all of the guidelines that you usually use for drawing, you will get your marks. And then now it asks for 2 similarities between a plant and animal cell. So even though it is a lab-based question, it still incorporates information that you would have learned in class. So it is kind of reinforcing what you would have done with that in a lab with other information that you would have learned from class. So what are the similarities between the plant cell and the animal cell? What do they have in common? Yes, Brianna. So both have a cell membrane.
Both have a nucleus.
You can say both have a nucleus. Oh, Kaylee, forgive me if I'm pronouncing your name wrong. Yes. Oh no. Yeah, they don't have cell wall. Cell wall is only for plants, so you can't ever use that one. Cell wall only for plants.
Both have cytoplasm.
Both have cytoplasm. Yes, Kevisha, that's what I was looking for. Both of them have mitochondria. The ribosome, I feel like you have to go so far. I feel like the four that are there are the safer ones to use. So go with things that are right on the spot. So you don't have to if I'm about it. because there is a lot of debate about the ribosomal thing. So it's better to stick with these ones. So both have cell membrane, both have a nucleus, both have a cytoplasm, and both have a mitochondria. If you put both of them have a ribosome, I don't think they would give it to you around, but stick to these. Any of these four should be fine for it. Yeah. Then number four, prominent structure on the animal cell electron micrograph. Again, guys, we just said that they are nucleus. Yes, Justin, why you put the question mark beside it? You know that's the answer. It is the nucleus. Again, the nucleus, whether it is an electron micrograph or it is under a regular light microscope, it is always going to be one of the most prominent structures. The nucleus always absorbs the most dye. Because remember that whenever you are making a specimen, if you guys had done the animal cell lab, you would have had to scrape inside of the cheek and then you would have dyed the cheek cells with methylene blue. So the methylene blue is a dye. It also is absorbed mostly by the nucleus. It is iodine as one. It is also absorbed mostly by the nucleus. So any dye that you're using absorbed most by the nucleus and it will be the most prominent structure. In the electron micrograph, it looks very dark again because of the kind of denseness of it, because it has so much material inside of it. It will show up with a lot of contrast. So it's going to look very dark. So it's always going to be the nucleus. And then even if you never know that, it says it carries genetic information in the form of DNA. And you know, so that's what the nucleus is. See, the CSEC people, they kind of make it easy for you in a weird way, both easy and both hard. Because sometimes they're there, they will give you the answer for one of the questions that they would have asked you further down in the question. So what you can try to do, read the entirety of the question, just run through it first, and then you start answering. Because sometimes they will give you clues. Because see here now, they tell you that it's an electron micrograph. So if you weren't sure about the question up here, we ask you what type of microscope was used. If you had gone down just a little bit, you would have seen it where it says an electron micrograph. So you can put two and two together and know that it would have been an electron microscope. So as a matter of fact, you see when you go into the exam, they usually have a clock in the exam room, and some of you have your watches on your hand. Use the watch or use the clock and time yourself. The first 15 minutes, you take it and you look through the paper and start jotting down in pencil everything what you know. Because sometimes when you start answering the questions, everything flies out of your head. So even if you had known the answer for question 6, because your brain done bust from question 1 through 5, you don't remember some of the things for question 6. So go through, take the first 15 minutes, and just jot down the information. A bit of information for every one of them so you can kind of understand the direction that you would have gone with your answer. Right? So those are just some tips for you what you can try to do to make it kind of easy on you, because as I said, they kind of make it hard, but they kind of make it easy as well. They will give you clues sometimes. Yes, Indiana School Challenge quiz. We're being serious about this. I want to know that you guys can actually answer some of these things, because if by now you have the exam on Thursday and you cannot answer, that don't look good. So we're just trying to go through as much as possible, and you giving me the answers makes that easier. So it's moving on. So figure 2. It says it shows the general structure of a bacterial cell. So it's asking for one similarity between the two of them. All right, Justin, I like how you never put no question mark beside the answer there. Yes. So both of them have a cell wall. Is it the same type of material that makes the cell wall though? Or are they made from different materials?
Miss, they're both genetic materials.
they both carry genetic material, but that would not necessarily be a suitable answer because the way that they carry their genetic material is different. In a plant cell, they would carry their genetic material in the nucleus, but in a bacterial cell, it's free DNA. So it is just condensed in a little space in the middle. So this structure that you see here is just their chromosomes. Remember that you cannot see the chromosomes when you look at a plant or animal cell because it is contained in a nucleus. But for a bacterial cell, it is different. Just so you guys know, in case they maybe ask you to label them, these are pili or cilia. Well, you can call them fimbriae. That's what they. So either pili or fimbriae, either one works. This area right here is the slime capsule. These little dots are the ribosomes. These are the flagella. And then the inner wall is the cell wall. So those are the general.
And the circles.
I don't remember what the circle is. I can't tell you what that is right now. It's something, but can't remember what it is. But these are the general labelings for it. The content of the cell wall is different. What is the plant cell wall made out of? Come on, bio students, what is it? Yes, cellulose. And then for the bacterial cell, it is murin, or you will see them call it peptidoglycan. So it is two different types of material that makes the cell wall, but both of them have the same. They both have a cell wall. So that's the similarity between them. Even though it is different materials, it is still the cell wall. We're good? Everybody up to date?
Mm-hmm.
Yes,
All right.
miss.
So this table now is asking you to basically give the benefit or the use of each of these structures in the bacterial cells. So the flagellum, anytime you see flagella or cilia or anything like that, they're usually used for movement. Locomotion. Slime capsule. Do you have an idea of what the slime capsule is used for? Or well, let me ask cell wall first. Cell wall first, and then we go to the slime capsule. What does the cell wall do in the plant cell? Because it would be the same thing basically.
Basically, select what enters and what go or what does not enter. Yes.
You're mixing that up, sweetheart. So the cell membrane is what controls the passage of materials in and out of the cell. The cell wall is just there for structure. So it keeps the shape and structure of the cell, right? Cell membrane is what controls what moves in and out of the cell. So remember, whenever you're talking about diffusion or osmosis, well, in this case, osmosis, it is the movement of water molecules from high concentration to low concentration across a semi-permeable membrane. Always remember that it uses the word membrane, so you can use that to link it back to the cell membrane to know that that is its function. So the movement of things in and out. That's what the membrane is for. Cell wall is just for shape and structure. So it maintains cell shape and structure. And then the slime capsule which is some might be new for some, but what do you have an idea of what it does? I think someone had started. Yeah, Kaylee. Sign capsule is for protection. So it basically protects it from a physical or chemical attack. So if it is obviously if you have bacteria, antibacterial stuff, or if you have antibiotics, it's the slime capsule that would kind of help to protect it from that kind of attack. So it protects physical or chemical attack. Because if you look at it again, you realize that it comes before the cell wall. So it is basically the first line of defense or protection for the bacterial cell, and then everything else would be the cell wall. So those are the functions of each of those parts. The bacterial information, it should be the first section of the book where they start talking about the different kingdoms. I think that is where you would find the information. So if you're not sure, you can go and try to read through that chapter and see the information that is here should give you. And then it moves on to ask you about your different types of cells. It says that we are multicellular and we are made out of a large variety of cells. Obviously, multicellular means that you have more than one cell. And then it is the opposite of unicellular, which is one cell. So in our case, we are made out of multiple cells, and we are given 3 of these different types of cells: red blood cells, skin cells, and nerve cells. And it is asking you for the differences in the structure. It is basically asking you to compare them. So if we're comparing a red blood cell and skin cells, what are some of the differences or the things that we can use to compare them?
The fact that the red blood cell have a biconcave shape while the skin cell doesn't.
Okay, so the shape is different. The skin cell is basically the regular scale that we would have. It's unspecialized. Red blood cell has hemoglobin. And the skin cell doesn't. The nucleus, red blood cell does not have a nucleus, while a skin cell does. So those are 3 differences. One, the red blood cell has a biconcave shape, while the skin cell does not have that kind of shape. Red blood cell has hemoglobin, skin cell does not have hemoglobin. Red blood cell has no nucleus, while the skin cell has a nucleus. Again, all right, so thank you. Is that Bob? Where you said that is a difference in function. So remember that structure just means what it is, how it is made up, while the function is what it does. So this is asking specifically about the structure. So you would not give anything that has to do with the function. So you don't have to talk about no function at this point. So it would only be the structure. How it look? What are the things inside of it? That is what you would use to compare them. And then, all right, so with that, remember you have to read carefully. A lot of the times it's not because you don't know the answer, it is because you have read it and you have not really paid attention to what it is asking you. You have to think about it. I'm not saying take 50 hours and sit down and look at a question, but you have to really think about what you're writing before. Because as a teacher, I can tell you that that is what I see, where students go in and they write the first thing that comes to mind when they're not really answering the question. And sometimes when I write out my heart and soul, and then when you look at it, you have not really answered the question. So please pay attention to what the question is asking you for. And then now, for this one, it asks for the difference between the structure of skin cells and nerve cells. So what are some of the differences that we could think about for those two in terms of structure? Anybody? Remember how nerve cell looks as compared to
Nerve
skin cell?
cells have nerve endings.
What are those nerve endings called?
Axon and dendrites.
Dendrites, dendrons or dendrites, whichever one. Extending from a main body.
Skin cells have pigments, melanin. That's in them. I don't even really know, but
Yeah. So I think the safest answer for this is the extensions of the axon from the axon to the dendrites and the those things. Whereas when you say that the nerve cell has that, but the skin cell doesn't. So that is the safest answer for this. So Kevisha's answer in the chat would be the answer that I would put on the paper. All right, and then it says, name one feature that would enable a scientist to distinguish between a spherical bacterial cell from an animal cell when viewed under a microscope. We had said, when I was talking about that, I gave you two that we could use.
The nucleus.
The flagellum.
The flagellum.
I'm so sorry. I was not hearing anyone a while ago, so can we repeat what was being said?
Bacterial cell will be the flagellum, I guess.
So a bacterial cell. Well, we could use it, but some bacterial cells do not have flagella. Some of them will have cilia. So we can say the presence of a flagellum or cilia.
The presence of a nucleus.
No nucleus.
The presence of mitochondria.
Well, animal cell has nucleus. For the mitochondria, you would not be able to see the mitochondria under, well, you could see it if it's an electron micrograph, but if it is not, you would not be able to see the mitochondria because they're going to be very small. So the nucleus is a safe option. Thank you, Kay. So the animal cell does not have a cell wall, while you know the bacteria cell has a cell wall. I think someone was saying the other one with DNA. Someone was saying something about it. Yes, Bob. Animal cells have nucleus, DNA enclosed in the nucleus, while the DNA strands are free in a bacterial cell. So a bacterial cell has DNA free in the cytoplasm. So you will be able to see for yourself as some little string-like structures, while for the animal cell, you can't see the DNA because the DNA is enclosed in a nucleus. All right. So those are some of the things that we would use. And then again, the animal cells lack flagella or cells. Well, we should be able to work with that, yeah. Right, that's good. And that is the entirety of question 1. Again, you will see that is worth 25 marks. So you could get a good amount of the answers for question 1. And then for question 2, we're gonna be talking about meiosis and mitosis. So this one is showing you meiosis and what is meiosis used for, or what are mitosis and meiosis.
Mitosis is a cell division that produces diploid cells, for example, cells of the body, while meiosis is a cell division that produces haploid cells, for example, gametes.
All right, good. So mitosis, although both of them are forms of cell division, there are ways that new cells are made. The outcome from both of them and their purpose is different. So mitosis is there for, it produces cells that have the full genetic information. So they have the full set of 23 chromosomes. So they have the full DNA, 46 chromosomes, so 23 sets. And the product is that you have 4 genetically identical cells. So all of the cells coming out of mitosis are identical. And this type of cell division occurs in all body cells. So whenever you're making new skin cells, all of your skin cells have to look the same. So obviously you want the same type of skin cell that was there before to be produced. So you would have mitosis occurring in those kinds of body cells in order to produce the cells that are of the same type. So you are given 4 genetically identical cells. And it occurs in all body cells. And they are diploid, meaning that they have the full set of 46 chromosomes. Versus now meiosis, which gives you 4 genetically different cells. And then it only occurs reproductive organs. To produce gametes. All right, and in this case where it is meiosis, all of them are coming from this cell up here, which is the parent cell. So it would give you 4 genetically different cells. So if you look, in order to complete the diagram, because that's what it is asking you to do, it is basically asking you to give both of them give you genetically, I think it is, I believe it is 4. If I remember correctly, it should be 4. I think both of them are 4. That doesn't matter. Both, just know say both of them are whatever amount of them are produced are genetically different. Yeah, both of them give 4. Mitosis and meiosis give 4 cells. The only difference between the 4 cells is that for mitosis, it is going to be 4 genetically identical cells, while for meiosis, it is going to be 4 genetically different cells. So, in order to complete this one, you would just really look at the chromosomes that are in the parent cell and basically rearrange them. So for both of the other ones, you see where this comes here and this comes here. So you cannot use those combinations again. So you would basically have to do something that looks different. So something like this, and then you use this one, and then because you want them to look like they are genetically different from each other. Is that making sense, guys? I hope you understand what I mean.
Yes, miss.
You just need to ensure that they look different. So because the other 2 combinations have been used already, you're basically using one from this set and then one from the other set to give you the other 2 cells for Q and R. So you would have gotten 4 marks from there where you would have completed that figure. And then it says to state whether the daughter cells produced by meiosis are haploid or diploid. Once it is meiosis, it is going to be haploid. And haploid basically means half. You can always use the first part of the word to kind of figure out what it is. Haploid, half. Both of them start the same. And then diploid, di means 2. So basically, you can know that it would be 2 cells coming together to make one for a diploid cell. And then for your haploid cell, it is just saying that it is half of the whole. So remember that humans have 46 chromosomes. With mitosis, you get diploid cells, meaning that all of the cells have 46 chromosomes. And then for meiosis, it's haploid. And the cells would have 23 chromosomes. It is necessary for them to have 23 because remember that each species has a specific chromosome number. For humans, it is 46. For rats, it might be 23. For donkeys, it might be 42. So you have those numbers and they have to be what you would get for a species. In order for them to reproduce, you basically have to cut that number in half because half of the information is coming from the father, half from the mother. So if you do not half it, you will not get a whole to put together. If you come with 46 chromosomes from the mother and 46 from the father, meaning you come with the full set, that number will not give you 20, it will not give you 46 when it comes together. It is going to give you, what's that, 90, 92. And if it is 92, it is not going to be a human being. I don't know what that is, but it will not be a human. So that is what we have to remember. So it's always going to be half a meiosis. And then we said that meiosis only occurs in the reproductive organs. So reproductive organs, if you want to say that it occurs in the ovary, that would be acceptable too. If you want to say it occurs in the testes, that would be acceptable as well. And then it says name the structure within the nucleus that contains genetic material, and this would be your nucleolus. We're good, guys?
Yes, miss.
Yes, miss.
Miss.
Yes, questions?
Miss, someone has their hand raised.
Shane, do you have a question?
Miss.
I'm listening.
Miss, the question that you said, name the structure which the nucleus which contains a genetic material. The structure within the nucleus which contains the genetic material, isn't it the chromosome?
I'm not hearing you.
Isn't the answer the chromosomes? For C.
Well, that could also be the answer. It's but the chromosome itself is not necessarily a structure. It's more like a content. So that is why I said the nucleolus.
I first missed the word.
Well, all right. So I guess we can go chromosomes.
Okay.
All right. So let's go with that one then. So either one. Well, all right. For your sake, go with chromosomes. It should be an acceptable answer. For question B, we're talking about observable characteristics, and they said despite them being siblings coming from the same parents, the observable characteristics are different, and you're supposed to use your knowledge of genetics to explain why the observable characteristics are different, even though they are sisters. So, what would be possible answers that we could use for this?
We could talk about meiosis and how the what's it called? The cells that they produce are different, as in they're not exactly the same, none of them. So the chances of the child like being born exactly the same are like
Are zero.
0.
So wonderful, great answer. So the first thing we could talk about is meiosis, and that we have 4 genetically different data cells produced. And because they're genetically different, every time you do it, because you have random distribution of chromosomes, right? So that is why we have the crossing over, and then we have the random assortment. So those are two things that happen in meiosis: crossing over and random assortment. Both of those will end up making the genetic material different from the jump. So it is impossible for them to look the same, because remember that the genes that you get would be different with each data cell. And your genes, which is your genotype, would affect your observable characteristics, which are your phenotype. So genotype is genes, and we get your genes from the DNA, and then your phenotype, observable characteristics. So the genes would be different for each data. Yep. So Alicia's answer in the group is a good answer for one. So that would be one reason. The genetic mutation, I would not. It could work as an answer, Joshua. So it's not necessarily wrong, but I would not personally put it here because even though we have mutations, I don't think the most acceptable answer for this part. So stick with the genes obviously being different because of um the random assortment and crossing over. If you are short pressed for another answer, you can't think of anything, then you can go with mutation. But in this case, stick with that one first. And then one other reason we could also say, I think someone else had said something, environmental factors. We could say that as well, because even though they have similar genes, they're both exposed. They can both be exposed to different environmental conditions. So that can work as well. Anybody else have any answer that we think we could put? Or are
Can
we
you
gonna
explain meiosis again, please? And how the genes would be different from each other for each daughter cell?
well, already that would have been the first reason when we said that both of them are genetically different because of random assortment, due to random assortment and crossing over. Yeah, Justin, that can also be a good one. Talking about genotype, the genotype could be different in terms of whether they got heterozygous or homozygous dominant or recessive, because that would also affect um their phenotype. So heterozygous. So we would those two answers or those three answers, as I said, if you can't think of anything else, you can always go the route of mutation. So those answers can work. And then we are saying, why is meiosis important? So two reasons why meiosis is important. The first one, we said it before, it maintains chromosome number.
Miss, if you're speaking, we can't really hear you.
All right, guys. Sorry about that. I got kicked off. So this whole time I was talking and I did not realize. So sorry for that. Just confirming, you're hearing me now?
Yes, Miss, we're hearing you.
Yes, miss.
Yeah. I'm not sure what's happening with you. Sorry, guys. Um, all right. And you're seeing my screen as well?
Yes,
Yes,
Miss.
miss.
Okay, cool. So we were at this question about meiosis in flowering plants, and we were asking name two of the gametes that are produced. The female gamete is the same, so it would be the ovule. And then what is the name of the male gamete that we use for yeah, so pollen. And the pollen contains sperm nuclei. Then we go down to question 3, which asks about the kingdoms. How many kingdoms do we have?
5.
So we have 5 kingdoms: animal, plants, prokaryotes, protists, and fungi. So we can give any three of those kingdoms except for the animal kingdom. If you can't remember any other one, you must can remember plants and fungi. So at least those you'll be able to remember. And then you can choose either prokaryotes or protoctis to remember, whichever one is easier to spell. So better you go with prokaryotes. So any of those outside of the animal kingdom you can use. For number 3, fish are organisms that belong to the animal kingdom. We have 6 species of fish that live on the coral reef, and we're supposed to use 3 visible characteristics that we can use to clarify the fish. So what are 3 of them? The first one that is very obvious. The first thing that you see about the fish.
The type of scales, pattern.
If they have gills,
All fish have gills, so you can't
The
use
size of their fins.
gills.
The size of their fins.
Okay. There's one word. I know that person is
Size of
actually
their
the
mouth.
first thing you see. Tail shape. That's the first and the safest option. Because we have this type of tail, and then we have this type of tail that we can obviously use to separate them. We could use scales, scale pattern maybe. We could use the, what are some people saying?
makes the number of
Shape,
fins
size
that they
of
have.
things. The number of what?
Fins that they have.
Number of fins, okay. Also, we can look at the specific type of fins that they have. So, for instance, this is a pectoral fin. This is a pelvis fin, and up here, this one, or these, or dorsal fins. So we can use presence of a dorsal fin, or shape of dorsal fin. We can use presence of a pelvic fin, or presence of a dorsal fin, because not all of them have one. So for instance, this C over here does not have a pelvic fin. E does not have a pelvic fin. So we can group them in a group by themselves because they don't have a pelvic fin, while F, B, A, and D have pelvic fins. So pelvic fin, we can look at their dorsal fin if it is joined or if it's not joined. The dorsal fin is what's on the top up here. So we can look at that as well. Dorsal fin. So those are some of the things that we can use. We could maybe use mouth shape. That's also one. Or mouth size. Those are good ones. So any observable characteristics that we can use to basically determine, put them into different groups. So any of these are acceptable. The most obvious one is this one, the tail shape. All of the other ones are also acceptable. So either one can work. Okay. And then we go down and it is basically asking you to group them. So going back to the first thing that you would have learned in grade 10 about grouping organisms or classification of organisms, where you would use a characteristic and put them into 2 different groups. So it says use any one of the characteristics to put them into 2 different groups. So for instance, tail shape. Okay, so this is kind of it's asking you to use 2, tail shape. I don't like the way this question is worded because I feel like it leaves room for misinterpretation. And I also don't like the table because it's slightly confusing, where you're unsure if you're supposed to use how you're supposed to do it. But it's because it says use any one characteristic, I assume that you're just going to use the two different columns for each of the groups. So for instance, if we go and we are putting them into the group now, and we're using tail shape. So all of them that have the same tail. So we would have group 1. And then we would have group 2. So all of them that have the same tail shape. So we would put F, C, and E in one group. And then we would put A, B, D in another group. Because when we look at their tails, it would be different based on how one of them is like it is cut in two, and then the other one is just a solid tail. Is someone trying to say something?
Yes, so instead of putting tail shape for both of them, we could put the different, or maybe the name of the tail shape. So say for instance, one has a crease and one doesn't.
This one over here, E, and when we look at that, that's a fan shape. I do not recall what this shape is. So I guess for the sake of simplicity, we just say group 1, group 2, and we just put it based on whether or not it's a fan shape or if it's not a fan shape. So that is how we would do the groups. If you were doing mouth shape, you can say the ones that have kind of an elongated mouth, so it would be F and C in one group, and then all of the other ones in one group by themselves. If we're doing the presence of a pectoral fin, E and C would be in one group, F, B, A, and D would be in another group. So any one of the characteristics that you're using, as long as they have the same one, or if it works, we would do those two. And then we go down, question C. It says define the term conservation as used in biology. So conservation is the process by which environment is safe or maintained. Maintained for future generations. In the simplest of terms or definitions. Does anybody else have another definition that they would like to use here, or are we okay with this? Nobody? Okay, I'm assuming. Okay. Sustainable use of natural environment in order to preserve it for future generations. That can work also, Bob. So that's a good one as well. But as long as you're saying that you are maintaining the environment for benefit of the future generation, that is the integral part for you to have in your definition for conservation. Anything else that you want to put as fluff, that is it. But the meat of the matter is that you're here to preserve it for future generations. And then we're moving on to number 2, where it's the fish populations that live on coral reefs in the Caribbean are under threat of extinction due to predation and pollution. So it's asking you to give 2 methods of conservation that may be used to minimize these threats. So I am assuming that it is asking you for one for predation and one for pollution. So you have to give one way to minimize pollution and one way to minimize predation. So pollution, predation. Predation is when you have different predators coming onto the coral reefs. They are usually invasive species, meaning that they are not native to the area, they're not here. they were brought here by something. Usually, when it is the coral reef, it is brought through the fuselage of um, the cruise ships or the tankers that come in with goods. They're usually taken up in one part of the ocean and then they get stuck in the fuselage and are released in the Caribbean waters. So that is how we would get those predators there. Okay, so Bob, your answer works. Where first of all, if it is pollution, once it is aquatic pollution, you obviously want to deal with fertilizers. So yeah, fertilizers is the big one that you want to deal with. So you could deal with limiting the use of chemical fertilizers as it makes its way into the sea, or you can just you can minimize the use of those kinds of fertilizers, which have the harmful chemicals inside of it, which obviously would lead to eutrophication. Or if it doesn't lead to eutrophication, it will change maybe the pH or certain aquatic conditions that are necessary for the survival of those organisms. So obviously, pollution, you want to minimize or limit entry of chemicals, whether it is fertilizers or pesticides, the runoff, chemical runoff.
Excuse me, miss, for predation, could you like introduce or increase natural predators for the predators?
So one of the ways that we deal with predation, we either use biological control, which is when you and that is the more dangerous part because you want when it is not native to the water, you usually have nothing that can eat it. So for instance, in our waters, we have obviously the lionfish is the biggest one, where it is an invasive species. It was brought here and it is what is predating on a lot of the organisms that are on our coral reefs. Because of that, we have no because they are invasive, meaning so they weren't here in the first place. So nothing here and I have no way to defend against them or to actually attack them for the most part. Introducing something that can get rid of them is posing the risk of them themselves becoming an invasive species. So then you have two invasive species to deal with. So biological control would not be the safest method for this. Not saying it can't work, but it will not be the safest method. Or would not be the most practical method. If you cannot think of anything else, then you can go with that method. But in this case, when it comes to predation, you're going to do something called culling, which is when you have local fish
Good evening, everyone. Can you indicate if you're hearing me, please?
We're hearing you.
Okay, great. So you would have noticed that your tutor was kicked off. She has been battling with the whole internet issues from last night. She relocated and she's back in that issue. So we're going to just hold on until about 6:30. If it doesn't come back in time, then we may have to reschedule that. Just know that anything that we have to reschedule will not be recorded. So that is a timing outside of NJF's time. So that is like a proposed Google Meet or something of the sort. And do you guys take it from there? Can anyone tell me when is the exam for this, please?
It's next week, Thursday.
Okay, so there's still some formal leg room for preparation. Okay, so as I said before, guys, we're waiting until 6:30. If she's not back on yet, then you know what that means. Chanel, is that the pronunciation?
Yes, it is.
Okay, great. I'm gonna kindly ask you to stop the recording for me, please.
Oh, okay.